we very often have lectures, conferences, and all start sharply at the time indicated in the program. So it's 9 o'clock, and we will start. Do you agree? Yes. Good morning, everybody. So we will continue our conference. And well, sorry that the weather is not the best these days. In September we had very nice weather, it was sunny, but for the conference sometimes it's good to have rainy weather. So the first presentation will be given by Professor James from the Yale University, United States of America. Let me just a few words about Professor Rotman. Professor James, Nobel laureate in Physiology or Medicine, 2013, is one of the world's leading biomedical scientists. His pioneering discoveries on how cells transport and deliver vital molecules, such as hormones, enzymes, and neurotransmitters, have fundamentally changed our understanding of cellular communication. Through decades of meticulous research, Professor Rotman uncovered the molecular machinery that allows vesicles inside the cell to fuse precisely with their targets, ensuring the accurate flow of life's essential substances. His work has laid the foundation for major advances in medicine and molecular biology. Born in New Haven, Connecticut in 1950, and now professor of Yale University, James Rotman combines scientific rigor with deep intellectual curiosity. His Bessarabian roots, with ancestors originating from Palești and Telenești in present-day Moldova, and a profound symbolic dimension to his visit to Chișinău, marking a return to his ancestral homeland after more than a century. Professor Rotman's legacy reminds us that science is not only a search for truth, but also a bridge between generations, nations, and the shared pursuit of knowledge. Thank you, Professor Rotman, for your visit. And you have now the floor. You see the title, the biophysical basis of rapid information transfer in the brain. You have the floor, please.
Well, I would like to thank you, Yuan, for a very kind introduction. This has been an amazing trip for my wife, Joy, and I. And I'm very much honored to be here today, also to be also as a guest of the Academy of Sciences in Moldova. So I thank you all for that. Those of you who live here, I thank you for the weather. That's a joke. What I'd like to talk to you about today is perhaps the most remarkable piece of nanotechnology and biomedical engineering ever made. And it is of humans, but not by humans. And I'm talking about the brain. And I would like you to think for a moment from an engineering or a nanotechnology perspective about the brain as an information processing device. And then, with that in mind, I'd like to ask a question with you. What sets the speed with which we can process information? And then I would like to drill down on that question and explore with you at a molecular, near atomic level, how that actually works. We don't have the answers to all of those questions, but the answers are coming rapidly through research in our laboratory at Yale and in other laboratories around the world. So there are, this diagram represents two nerve cells, or neurons. One is in an instructive mode and the other in a receptive mode. These nerve cells have, of course, cell bodies with nuclei. They have extensions, which I'm sure you're all familiar with, called axons, which are like wires. And then these wires contact each other, and the contacts between the cells, between the tip of the wire, called the nerve ending, and the cell body of the next nerve, these are called synapses. And these synaptic connections in the brain are very numerous. This is where information processing is taking place. And so you have an action potential, which is a form of electrical conduction that goes down the nerve axon, which has underlying it the motions in and out of the cell of sodium and potassium ions. I won't be talking about that. And then you have the transmission of the information from that signal, from one cell to another at a synapse. And that transfer of information, as I'll describe, is chemical in nature. And the chemical that transmits the information is what we know of as neurotransmitters. Okay? So those are the basics of the field. And information flows in this direction. It's one way. And then, of course, this cell will connect to another cell. And the pattern of those connections will represent the circuit. Okay? Now, an important feature of brain anatomy, not just human brains, but all nervous systems, is there are an enormous number of wires, but a still larger number of connections. So this is a reconstruction from recent work in so-called connectomics of about a cubic millimeter of the human brain. And what you're looking at here are just the wires that are present there. And what you can see from this is that there are many wires, yes, but they're also very short. On the other hand, in the entire brain, or in a region like this, in the entire brain there are actually about 1 billion axons, meaning 1 billion wires, but there are 10 to the 12, 1 trillion connections. 1,000 connections for every wire. So if you wanted to think of our brain as a neural network, it would be a highly interconnected neural network. And so it should not surprise you to know that the vast majority of energy used by a brain is used at the synapses, not in these short wires. And it is the synapses that have also the unusual property of storing memories. They can store memories because the strength of each of these synapses is individually adjustable. It can be adjusted according to how frequently it's used, and once it's adjusted, the newly adjusted strength is a constant. And yet it can be changed over time. And so this is an important aspect of how memories are stored. And so what I'd like now to do, with this in mind, is ask the question, what sets the speed of information processing in the brain, the speed of our central processing unit? And I think you can now see that the speed is going to be set by the speed of these connections, because the vast majority of activity is synaptic activity. So let's take a closer look at a synapse. So this is now, what we're doing is looking at this area right here, in which the upstream cell, the upstream nerve, is connecting and transmitting information to the downstream or postsynaptic neuron. Presynaptic, postsynaptic. And when we take a look inside of that box, we see is the structure of the synapse. And a synapse, you see, is a highly polarized structure. On the presynaptic instructive side, there are a large number of very small vesicles. This is, of course, a cartoon. This is an actual electron micrograph of what it looks like. And here's the presynaptic side, and you can see many little vesicles. Each of these vesicles is a packet of information. It contains the neurotransmitters. There are about 5,000 individual neurotransmitter molecules, one identical to the next, that are present inside each of these vesicles. And the vesicles are called synaptic vesicles. They were discovered, actually, in the early 1950s by George Palade, an earlier recipient of the Nobel Prize in physiology or medicine, and also by Sir Bernard Katz at University College London, who discovered the nature of these quanta of information in synaptic transmission. Well, what happens then is these vesicles are released by a process called membrane fusion, in which the membrane enclosing the synaptic vesicle merges with the outer plasma membrane on the presynaptic side to release the neurotransmitter into the space in between, termed the synaptic cleft. And then there are receptors on the other side, the postsynaptic side, over here, of the next nerve cell that bind these neurotransmitters. And when they bind the neurotransmitter, they open up ion channels. And the ion channels serve to depolarize the cell body of the receiving cell. And if there is enough input from enough synapses that depolarize the postsynaptic cell, then that postsynaptic cell in turn will fire an action potential. And so you can see then how each synapse, depending on the nature of the connections, the number of the connections, and where they come from, makes a decision of whether to fire or not fire. That's a binary decision. Likewise, it turns out that in the central part of our nervous system, a very small number of vesicles will fuse at each synapse. In fact, it's only one. So either one fuses or zero fuses, and that probability is actually the strength of the synapse. So what you have, what I'm describing to you, is essentially a highly complex digital computer that transmits information at the synapse in an all or none fashion, and that makes decisions at the level of the postsynaptic cell to fire or not to fire another action potential, to pass a signal down. And it's so it's not a bad model to think of us, our brains, as digital computers. Now, I'd like to take now a broader, so what I'm going to be addressing then is what is the molecular mechanism by which these synaptic vesicles fuse to release their neurotransmitter, because that is the determining event for information transmission at a synapse. And as I've said, that event is probabilistic. It can either happen or not happen, and how frequently it happens essentially is the strength of that synapse. Okay? There are other components to synaptic strength, but that's a key component. And so there must be a molecular machinery underlying this that somehow allows that to happen. Now, there's another element of the machinery that underlies synaptic vesicle fusion, which is the speed. Because if it's slow, if it's too slow, then our processing of information will be correspondingly slow. In fact, it's remarkably fast. And it's that feature, the speed of this process, that requires a singular explanation that I will offer you today. But first, let's look at synaptic transmission in a broader context. Let's look at it in the context of membrane fusion generally. Because vesicles, membrane vesicles, of course, are found in the synapse, but they're also found throughout the body. Insulin in our pancreas is stored in secretory vesicles, as are digestive enzymes in other portions, the exocrine pancreas. Inside of every cell is literally teeming with many vesicles that carry with them proteins that are traveling from where they're made in the endoplasmic reticulum as part of cell growth and division to whatever organelle, including the cell surface, they reside in. That process actually was discovered by the Romanian scientist, George Plenary, who is actually my predecessor at Yale in leading the cell biology department. And what we discovered that resulted in the Nobel Prize in 2013 is the general principle that underlies all of these vesicles, whether they are involved in cell growth and division, the secretion of hormones or development of cell tissues, which also rely upon local hormonal secretion, or in the case of the brain, the transmission of information by synaptic transmission. And that general principle involves a class of proteins we discovered, which I call snare proteins. And the snare proteins exist in two types. One type here in blue is in the vesicle, and the other is always in the membrane with which the vesicles fuse. And it's actually a very simple idea. The proteins, the snare proteins in the vesicle partner up at the molecular level with the snare proteins that are coming from the plasma membrane, and that partnership takes the form of an alpha helical bundle. That alpha helix is very strong, and as it assembles, it pulls the two membranes together. And when it does that, that force literally pushes the membranes together, the two lipid bilayers, and they are fused. So it's a very, very general principle. And you can imagine in physiology that force of the snare proteins that can be used to fuse a vesicle with a membrane can be triggered or harnessed in many ways. So there are many ways in which that force is regulated in biology. And the regulation differs from cell to cell, from one part of the body to the next, according to the physiological need. So for example, in cells that are growing, you know it takes a day for a cell to double its size. So correspondingly, the process of membrane fusion can now take place on a time scale of seconds. On the other hand, we know that hormones like insulin can be released quite rapidly, and they need to be. And they take typically about a second. So when insulin rises in the blood, those of you who have medical backgrounds will know that your blood sugar responds very quickly. And the first phase of insulin secretion actually takes place within about 1/10 of a second to a second. But what you may not know is that synaptic transmission takes place 1,000 to 10,000 times faster. That is really remarkable. It takes about 1/10 of a millisecond to 1 millisecond, meaning 1/1000th of a second or less, for this vesicle to fuse, to release the neurotransmitter, and for the neurotransmitter to activate these channels. If it were any slower than that, our brains would literally be slower than they are. And our species would be stupider than it is. Now, one of the things that those of you who focus on politics and government and international issues will understand is it's hard to imagine the human species being stupider than we are. But we could, in fact, be stupider if this were slow. Now, amazingly, this speed is really fundamental. A fly or a worm, they also have synaptic transmission in their humble nervous systems. It also takes place at this level of speed. Why? Because this process of synaptic transmission is so highly evolved that it has not changed in all of those years between the fly and the worm and the human. It has not changed. Because the need to survive has not changed. And the law of survival of the fittest has not changed. And indeed, you will come to see that the speed has evolved to the limits of physics. The speed is set and limited by the rate of physical diffusion of proteins in a membrane. So it cannot be faster, and our brains are evolved to the limit that physics allows. So what I'd like now to turn to is the question, how can the speed of this memory fusion process be so very fast here when it relies on the same proteins? You know, I'm telling you that the same engine, snare proteins, is used here, or here, or here. If I told you that your car engine, the same engine, could run at 1,000 RPMs, you would say, yeah, no problem with that, okay? Whether you have a Skoda or a Mercedes. But if I told you that the same engine could also run at 10 million RPMs, and it would be the same engine, you would say, that's crazy. How could that be? And so that's the question that I've been focused on for the last several years, is how can the same system of snare proteins vary in the rate over such an extreme degree? And what I've come to the conclusion, and it's more of a hypothesis than a conclusion because we're still testing it, but I've come to the conclusion that it's very likely that the answer comes from the organization of the proteins that regulate the snare proteins. So I now have to make things, I've tried to make things very simple. Now I have to unfortunately make things a little more complicated because reality is always a little more complicated than our diagrams. And now I want to look at what the synaptic vesicle actually is made of. First, I want to tell you what its size is. The diameter of a synaptic vesicle is about 40 nanometers. Okay? Just to give you a sense of scale, that corresponds to 400 hydrogen atoms. The hydrogen atom is probably the smallest atom. And if you take 400 of those and you line them up, one next to each other, that's the diameter of the synaptic vesicle. So it's almost on a molecular scale. And in the spirit of this conference, I believe we would describe it as the nanoscale. And so let us think of this as a nanotechnology with extraordinary performance characteristics. Inside of that, again, are the neurotransmitters. Now, the protein components of the synaptic vesicle are by now well understood. And of course, there are the snare proteins. And this particular snare protein has a name that doesn't really matter. But this is the V snare, or the vesicle snare, that I alluded to before. Now, in the presynaptic plasma membrane, the nearby plasma membrane at the nerve ending, there are two snare proteins. Again, they have names, they don't matter. SNAP25 and syntaxin. And it's these three proteins, this one, and this one, that will partner with each other to form the pin-like structure that will fuse the vesicle. Now, the rest of these proteins are there for the purpose of controlling that reaction. Because the snare proteins will assemble by themselves. They don't need any help. But most of these proteins actually prevent them from reacting. And then you actually release the inhibition at the right time. And that's how they're regulated. So these proteins are what are called molecular chaperones. They chaperone the snare proteins. They're bound to them. They prevent them from interacting with each other, just like a human chaperone might, in the old days, prevent a young lad and a young lassie from interacting with each other. And so too, there are chaperones here. And they have many molecular details, and there are several of them. But the most important one that I'll talk about is this one, called MUNC13. And it doesn't matter where the name comes from, but I'll give you an idea of the history of the field. M stands for mammalian. UNC stands for uncoordinated. And 13 stands for gene number 13. This was discovered originally by Sidney Brenner, the Nobel laureate who paved the way to understanding the structure of the gene, and later the structure of the nervous system in the worm C. elegans. And he and his colleagues identified a number of mutations which disrupted the function of the nervous system of the worm. And what they found was that gene number 13 disrupted the release of neurotransmitters. In later years, others, especially my colleague Tom Suthoff, with whom I shared the Nobel Prize, discovered the nature of the protein that was encoded by this gene, and then the structure of this protein. And so we actually have the three-dimensional structures of all of these proteins that regulate it. But what's special about lung 13 is its size. This is actually its structure drawn to scale. You see it's nearly as big as the vesicle itself. And it plays a pivotal role. Number one, it chaperones the assembly of these 3 snare proteins together. You need it for them to be brought together. Secondly, it actually binds the vesicle and it binds the membrane at its 2 ends. And as you'll see, it actually assumes a number of molecular states, digitized states. I refer to it as a ratchet. And it grabs the vesicle and it ratchets it down to the membrane in a series of stages. And in the process of ratcheting, as you will see, this exquisite machine is assembled. And so there are then a number of these proteins that are important for the bringing together of the snare proteins. And there are yet other proteins that I won't go into that arrange the vesicles on the surface of the membrane together with calcium channels, because an important point I need to make is that the calcium ion is actually the actual signal that triggers the release of the vesicle. I forgot to make that point. When the action potential reaches the end of the nerve, it opens up the voltage associated with that action potential. Literally opens a voltage-gated calcium channel that admits calcium only in this region. And so, and when the calcium ion enters, it triggers this process of release. And it's then, from the moment that the calcium ion enters, we can start the clock. And it takes less than a millisecond for the neurotransmitter to diffuse. Okay? I hope I'm being clear. Now, what I'm going to tell you today, I'm going to check the time to see how I'm doing. Not super well, I can see. What I'm going to tell you is that the amount of energy, the snare proteins represent energy. And the energy is stored in a container. And that container is triggered by calcium to release. And just like in the champagne bottle, that energy is stored. So if you think of the champagne bottle as the vesicle, and if you think of the cork on the champagne as the vesicle, as the interface of the vesicle, and you think of the calcium ion as releasing the cork, then the neurotransmitter, which is the champagne, is released in an explosion. And I will illustrate how that might work physically. And so our hypothesis is, essentially, that if you store enough energy, the snare proteins, in doing work on the membranes, are imparting as they assemble, they are imparting potential energy to those membranes. That potential energy is being stored in the vesicle and the membrane at the interface in an elastic way, if you think about it physically. And then, if you release that energy abruptly enough, then you will have an explosion. If you think about the nature of an explosion, okay, you can have, for example, gunpowder on a piece of paper. You can light the gunpowder, and what will happen? It will burn, smoke will come off, but there's no explosion, right? But if you take that gunpowder and you put it in a very small volume, and you give it a strong wall, okay, now you will accumulate the pressure associated with the gas when the combustion takes place. And it will accumulate until the strength of the wall is overcome, and there will be an explosion. And explosions occur at a speed much higher than the rate of diffusion. And that is why we think that there's underlying the speed here, essentially an explosion. And now I'm going to be running out of time, so I'm not going to be able to show you a lot of data. So I want to just tell you a little bit about our conclusions, and I'll give you a flavor of the data. And so the source of the energy, as I've already mentioned, are snare complexes. The snare complex assembles between the two membranes, pulls the membrane together, and in so doing, before the membrane is released, that energy is stored. And what we've discovered in the last 2 or 3 years is that there's a very specific number of snare complexes that assemble under the vesicle. It's actually 12, not 6, not 9, but exactly 12 snare complexes. This is a machine that assembles that has a definite stoichiometry, and as you'll see, it has a symmetrical, almost crystalline structure, even though we don't yet know the high-resolution structure. These snare proteins are assembled actually in two rings. There's an inner ring, which we call central snare pins, and then literally an outer ring of peripheral snare pins. And they're held in place by some of the other proteins that I mentioned. But there's an inner ring and an outer ring. And the inner ring, the central ring, assembles first and energizes the vesicle to a certain degree. And then, as the vesicle gets closer to the membrane, the next set of 6 snare complexes assemble. The result is a huge amount of stored energy. We actually know how much energy is stored in each of these pins because we can melt the pin and in a calorimeter measure how much heat is released or how much heat it takes to melt it. So we actually have numbers here. And the answer is 1200 Boltzmann constant kT worth of energy is stored in this vesicle for each vesicle. And for those of you who are familiar with biochemistry, ATP is the common energy currency in the cell. The strength of an ATP molecule in energetic terms is approximately equal to that of a covalent bond of an organic molecule. Why? It takes one ATP to synthesize or drive the synthesis of a typical carbon-to-carbon bond. And in the metabolism of a carbon-to-carbon bond, when we eat food, there's one ATP worth of energy typically that's released. There are about 100 ATP equivalents of energy stored under each vesicle in a thickness that is about 1 molecule thick, because the ready-release vesicle, which is the vesicle that I'm referring to, that is released by calcium, it's separated from the plasma membrane by only about 4 nanometers, which means 40 hydrogen atoms, 1 protein molecule. So now let's think about that. The energy is a high amount of energy is progressively stored in a smaller and smaller volume. Does that sound like pressure being built up? Energy in a smaller volume is higher pressure than the same amount of energy in a larger volume. Think about a gas. If you compress it, its energy level goes up. It's just basic physics. Now, the chaperone, monk 13, initially captures the vesicle in an upright form. It then changes its positioning on the membrane, and it forms a 6-member closed ring. Each of these structures can be observed in the electron microscope at high resolution, and we've done so. And as this vesicle is then, as this molecule rearranges, can you see that the vesicle will be pulled towards the membrane? It then rearranges into another structure, another ring. We don't yet have the structure of that ring. We're working on that now, but we know that it exists. And it is in so doing, monk 13 pulls the vesicle even closer to reduce the volume. And so in the first wave of monk, remember, monk 13 assembles the snare complex. So I'm describing to you a container. Doesn't this look like a bottle? And this is the cork, okay? So this is the bottle, and the wall of the bottle is assembling the snares inside of it. Therefore, it is a container. The container self-assembles. This is an amazing nano machine. It then, within itself, stores the energy first in 6 snare pins, then in another 6 snare pins, and it stays there stored until calcium ion enters. And in this way, neurotransmitter is only released when you have a signal coming from the upstream nerve. No neurotransmitter is released until the action potential leads to the entry of calcium. I hope that's clear. That's why we're transmitting information, not noise. Okay? And then this step occurs in 100 microseconds to 1 millisecond. It's so fast we don't actually have a great measurement of it. Now there's a third device which is equivalent to the cork, quite literally, and that is the calcium sensor. And there's a protein, again this one was discovered by Tom Tudor, the gentleman I mentioned earlier. And this protein is the actual sensor for calcium. The protein initially comes from the synaptic vesicle, but it binds to the plasma membrane, and as we've discovered, it assembles to form a circle, a ring which has 18 members. And that 18-member ring is assembled in the absence of calcium, and it sits right here. And it actually prevents the vesicle from fusing. But when the ring binds calcium, it falls apart. And when it falls apart, it fails now to block the fusion, and the fusion immediately takes place. So we have, therefore, three components that are needed for explosive release. A high amount of energy in a very small amount of volume, and a trigger that leads to precipitous release. Now this is an example, I just want you to see here, of the ratcheting process. The container is digitized. It is a molecule that can interact with itself in several different ways according to a highly evolved molecular program. And we are now obtaining high-resolution cryo-electron microscope-based structures of this protein, MUNC13, in the different molecular states. And what we're astonished to discover is that there's a limited number of these states that represent intermediates in the movement, in the drawing in of this vesicle to capture larger and larger amounts of energy. Okay? And here you have, I'm going to now show you the transition between this molecule capturing a vesicle and the molecule bringing in the vesicle. That's right here in this video. This is the upright form of MUNC13, capturing a vesicle, and you see how actually it can stay fixed on the plasma membrane and just undergo a simple molecular rotation. Look at that. And it just closes on itself. What a machine that is. Isn't that amazing? And this is going on in every one of your nerve endings all the time, and we don't even know it. This was only discovered in the last 2 or 3 years. Now, perhaps remarkably as well, and we'll try to finish up in the next 2 or 3 minutes, we've been able to reproduce all of this in the test tube. And that's how we know about it. And so what we've learned how to do, my colleagues and I at Yale, is to produce synthetic synaptic vesicles and synthetic presynaptic plasma membranes. We do this with pure proteins and pure lipids. And so some members of my team take the genes that specify MUNC13, as well as another key protein in the membrane of the vesicle, called synaptophysin, and of course the snare proteins, and the calcium sensor, synaptotactin. Altogether about 10 proteins currently. And we have 10 test tubes, each with a pure protein. And then we used technology that was available to us to formulate these into small vesicles in the same size range as the synaptic vesicle. I'll call those synthetic synaptic vesicles. And we can also make a much larger, flatter membrane formulation that mimics the plasma membrane. And it doesn't have all the proteins of the plasma membrane, only the ones that are necessary for the synapse to function. Okay? And we can lay this out on a slide for a microscope, and we can add the vesicles to it in this sort of test tube situation, all the while looking at a microscope at high resolution, a light microscope. And we can add a chemical dye, for example, to these vesicles. Or more importantly, we can fill the vesicle with a dye which is about the size of a neurotransmitter molecule. And when that is done, we can look to see if this vesicle will fuse with that membrane to deliver the dye, mimicking the neurotransmitter, to the other side. And we can actually visualize that in the light microscope in real time. And when we do that, this is an example. So here, we're looking at a single vesicle. There are hundreds of vesicles in our field that are attaching to the membrane. But I'm just showing you the signal from one. We can quantify that signal, and you see the vesicle has landed, just like the Apollo moon lander has landed on the moon. And it stays there. This signal is coming from the fluorescence of the neurotransmitter-like dye inside of that one vesicle. And now, when we add calcium, look what happens. The vesicle releases it. How do we know that? The signal actually goes up because the dye, at a high concentration within the vesicle, it quenches its own fluorescence. So as soon as the dye is released by fusion, that quenching goes away as the dye is diluted, and the signal actually goes up. And now you can look at the entire population of vesicles, and you can see here we have 2 milliseconds optical resolution. Within 2 milliseconds, the vesicle in the test tube has released, reproducing the extreme speed in the brain. Now, another thing that we can do, and I will be brief here, everyone, so don't have a panic attack, is we can measure individual molecules. So we can put our dye, instead of inside the vesicle, or in addition, we can put it on, for example, a snare protein. And when that snare protein is assembled under the vesicle, then there will be a step increase in fluorescence. And remarkably, this technology enables us to see single molecules, and therefore to count them as they arrive. And now what you're looking at is the same vesicle, but you're looking at individual snare proteins that are accreting to the vesicle. You see there are 2 waves. First there's 6, and then there's another 6. And we can measure that very precisely. So I did have a lot more that I wanted to show you, including what controls the number of snare proteins, and some of the molecular pathologies that occur in humans as a result of alterations in these pathways. And I was going to show you a precise molecular model, but I won't. And what I'll do instead is just leave you with the idea that there is a structure to be determined. I will show you one thing here. You are looking here at an electron microscope image of a synaptic vesicle at an actual nerve ending in nerves that are in culture. And what this is a synaptic vesicle looking from the top, looking down on it. But it's not one synaptic vesicle. It's thousands of them that have been averaged in a computer in an unbiased way. And when you do that, you can pick out tiny signals that would otherwise not be seen in a cryomicroscope. And do you see what pops out of it is a ring-like structure? These protein blobs are probably the elements of the machine that we've discovered in the test tube. You see that there are 6 of them, and each of them contains 2 snare complexes, as well as these chaperone proteins. We don't yet know how they're arranged, but we know that they're there, and therefore there's a structure to determine. And what my colleagues and I are working on, we have our ideas how it works, but what my colleagues and I are working on is how this particle forms, and at the atomic level, what exactly its structure is, so that someday we will be able to explain, hopefully not in the too distant future, the molecular base, the detailed molecular basis of information transfer in synapse, storage of memories, and the patterns of use of synapses that are likely altered in different states of physiology, such as learning as compared to thinking, and pathologies including neurodegenerative. So I thank you very much for your attention.
Thank you, Professor Bertrand. Thank you for the wonderful presentation. Ladies and gentlemen, the paper is open for discussion. If any questions, please. Yeah, please.
Thank you for the presentation. As part of artificial general intelligence, one of the questions which I guess comes up fairly frequently is how likely we can read the neurotransmitters using brain-machine interface, and I know interpretation of that information. We can read that because it's all
Yes, I
simple.
follow that work.
How likely you think, in your opinion, we can interpret that information?
You know, the whole question of the relation, let me generalize that question. It's a really, really good question, but I'd like to generalize it just a little bit. And we assume that artificial intelligence and the anticipated artificial general intelligence will create something similar in capability to a human brain. And I think that's the underlying assumption. I personally think we're very far from that. In the sense that the AI that we have today already does amazing things, but it doesn't come close to the sort of systems that we benefit from. Probably for dedicated purposes, it might be better. There may be cases, but the level, just energy efficiencies. You know, I think we have so much to learn from neurophysiology, neurobiochemistry, and even neuroanatomy in the form of connectomes. That will be useful in improving artificial intelligence. Consider the energy utilization of a human brain. For our brain to operate requires 20 watts of energy. Okay, so for you and me to engage in this conversation, if we were to talk for an hour, which we will not be allowed to do, this would consume the same energy as operating a 20-watt light bulb. You would have your light bulb, I would have my light bulb, 20 watts. Think about that compared to the amount of energy needed to train a neural network. Now, during that hour, I could learn many things from you, and you could learn many things from me, right? But if I want to train a neural network to do a fraction of what you're capable of, or what I'm capable of, I have to use the energy of 2 or 3 nuclear power reactors for a year. That's why Microsoft recently actually purchased the nuclear reactors at 3 Mile Island in Pennsylvania, which had been decommissioned for 50 years almost. 50 years. It's extraordinary how much more there is to learn.
Thank you, Professor. One more question. Professor Anthony, but please come here because you should use...
So I think I can speak loudly.
No.
Whatever he wants to hear.
Yeah.
So this is the delicate balance and equilibrium of the ions, the proteins, and all the lipids. And I noticed you mentioned PIP2 there.
Louder.
Normally the lipids really do play quite an important part. How do you control that in your reconstructed system?
Yeah, I obviously couldn't go into the details of it. PIP2 and diglyceride, its product, hydrolysis product, are both critical. It turns out that synaptotagmin, the calcium sensor, anchors to the membrane by PIP2 and therefore regulates that process. It turns out that the molecular switch between the upright conformation and the lateral connected conformation that I described, we call that lateral hexamer, that is governed by diacylglycerol, and it causes a molecular rotation of you'll be familiar with this, of the C1 unit of the attachment of monk 13 involves tandem C1 and C2 domains. C1 binds diglyceride, and when it's present, it drives that molecular rotation. So, I mean, the PIP2 hydrolysis cycle, which is itself, as I'm sure you know, governed by calcium, but on a different time scale, is involved in this. And so what you really have is an assembly line of vesicles that go from unprepared to the most prepared state, and 3 different time constants that are calcium dependent. You have the fastest time constant is the release of the vesicle, and that's essentially physical in nature, and that's synaptotagmin. Calcium also drives the disassembly of the calcium and diglyceride. Diglyceride in the absence of calcium causes the lateral hexamer to form. And then the collapse of that to make the ready-release vesicle requires diglyceride plus calcium, but lower concentrations of calcium than release, because that vesicle will be further from an ion channel. So there's a lot of details that are underlying this that we actually have elucidated.
Yeah.
Very good question. Thank you.
Yeah, thank you so much. Actually, amazing. I would like to say that your wonderful presentation raises also many questions and probably we will continue discussion because our brain consumes only 10-12 watts energy, but so complicated complex processes in the brain happen. I was really impressed by self-assembly processes, but we studied self-assembly, self-organization in nanotechnology, but in another field, of course. Dear Professor, so I have the privilege to give you this small award It's for developing and promoting nanotechnologies and biomedical engineering in the world and in the Republic of Moldova. Through your presentation, your visits, of course, you assist us a lot, and this was the decision of the program committee, the organizing committee of the conference. So this is for you.
Thank you.
Yeah. Okay.
Very good. Thank you.
We will wait for your next visit in Moldova.
Very good. Thank you.
Congratulations. So, there is a similar award. Professor Anthony Watts outstanding merits in developing and promoting nanotechnologies and biomedical engineering in the Republic of Moldova. I would like to know that Professor Watts contributed to the success of our summer schools. And we were happy, but I would like to say that students were more happy. Thank you so much. And this is the document. And this is actually the award.
Yes.
And there is one more issue. So, for his contribution to the success of summer schools that each year we organize at the Academy of Sciences of Moldova, there was a decision of the Presidium of the Academy. The decision was actually on 15th of October 1 year ago, but we had no opportunity to give you the award. So, certificate Dmitry Kantemir Medal is awarded to Professor Anthony Watts, University of Oxford, United Kingdom, President of the International Union of Biophysics, for outstanding contributions to the development of science, collaboration with the academic community of the Republic of Moldova, and for his contribution to the success of scientific conferences and summer schools organized by the Academy of Sciences of Moldova. Please keep this one also.
Thank you very much. Oh, thank you very much. No, I feel very honored, I have to say, and humbled, I have to say, by the previous recipients of the medal. It's been awarded since 1966.
Very nice. So,
thank
just
you.
keep it.
Yes.
Look at it. Congratulations.
Thank you very much indeed. This is so embarrassing. Thank you very much. Thank you.
I know we have.
Honor is awarded to Professor Rainer Adenu for outstanding merits in developing and promoting nanotechnologies and biomedical engineering in the world and in the Republic of Moldova. Professor Adenu, please come. Professor Adenu is among professors that attended all 7 conferences, with the exception of one. You attended online.
Yes.
But attended during the pandemic. Yeah. So this is for you, and this is for you. And please look to. We wish you much success.
Thank you.
Oh, sorry. Oh, Diploma of Honor is awarded to Professor Vladimir for outstanding service in development of nanotechnologies. So Professor Fomil is also among the experts that he attended all 7 conferences on nanotechnologies and biomedical engineering. Professor Fomil, this is for you, and this is
Thank you very much. It's a great honor for me to get this high decoration. Thank you.
Yeah, so the same award to Professor Toru Aoki from Japan. Is he in side note? Then we will take the possibility, maybe later we can give the award. So, Diploma of Honor is awarded to Untitled Chapter. I would like to thank you for your assistance, for your contribution to these conferences. This is your and this is your, and look at the... Ladies and gentlemen, it's my great pleasure to announce an award for Professor Victor Șontea, who is actually absent due to the fact that he has some problems, health problems, but he had an outstanding contribution to the organization of the 7 editions of the International Conference on Nanotechnologies and Biomedical Engineering. Of course, I will ask my colleagues to pass on this award to Professor Victor Șontea. So, ladies and gentlemen, thank you for the presence during this session. And I would like to point out on the line that moderator from this moment will be Professor Viorel Nacu from the State University of Medicine and Pharmacy, Nicolae Testinițanu. Professor, please take the floor.
We will continue our session. Sorry, I need my watches. It's my pleasure for me to ask Sheriff Comusciu to present the personalized medicine in cancer care, how genomic profiling and liquid biopsy are shaping the future of targeted cancer therapies. Please.
Good morning. Dear Chairman, dear colleagues, it's my honor to be here among those distinguished scientists in a so unique conference. I am a medical oncologist working in Anadolu Medical Center in Turkey. And I would like to thank to Professor Victor Chante and Professor Ion Tiginyanu and organizing committee for inviting me to this excellent symposium. I will talk about the precision medicine in oncology and genomic profiling, liquid biopsy, and targeted therapies, how they shape our oncology practice without overwhelming you the clinical details. These are my outlines. I will talk first about the cancer development and genetic changes in cancer development, and the precision medicine, and genomic profiling, liquid biopsy, the impact of targeted and immunotherapies, the challenges of precision medicine, and later I will complete with the future perspectives. As you know, cancer is a disease of DNA, and the genetic changes, alterations, mutations in DNA cause cancer. When enough mutations accumulate, the cell starts growing uncontrollably and cancer develops. Treating cancer is a challenge, you know, because cancer is not a single disease. It defines many diseases at the molecular level, each with its own fingerprint. Even if two people have the same cancer, their diseases may behave differently, and their responses to the treatments may change. So each patient with the same cancer type may require completely different therapies tailored to their tumor's unique genetic fingerprint. These genetic changes emerge precision medicine. Precision medicine involves customizing therapy based on the patient's or the tumor's genetic profiles. Instead of treating patients as averages, we now tailor therapies to the genetic profile of each tumor. This individualized approach, called precision medicine or personalized medicine, and these treatments have evolved from one-size-fits-all to more personalized, precise, and targeted treatments tailored to each tumor biology. Shortly, precision medicine is giving the right drug to the right patient at the right time. Recently, our clinical approach, the treatment changes a lot from chemotherapy to precision medicine. And the conventional chemotherapy is like using a hammer. It hits the nail, but also harms the environment. It is effective, but not so precise. Targeted therapy hits directly to the cancer cell while sparing healthy cells. Because of that reason, we don't see so many side effects due to targeted therapies. For precision medicine, genomic profiling is needed to reach cancer's fingerprint. For that fingerprint, we need tumor sample. It can be taken from the direct tumor tissue or from the blood. It is liquid biopsy. Then, the sequence of the tumor's DNA is needed. Next generation sequencing, NGS, is a comprehensive analysis of genome, and by this way we reach the genetic mutations of tumors. Bioinformatics tools involve the use of computational algorithms and software to analyze biological data. We reach the mutations driving the cancer, and that this information guides us to select the specific drug to each different cancer. The turning point for precision medicine is Human Genome Project. As you know, Human Genome Project is concluded in 2003. After that, genomics area has started. And the NGS test has developed, and the tumor genomes can be analyzed quickly and inexpensively. And we can reach the targeted therapies. One of the exciting innovations is liquid biopsy. It is a blood test, and it detects cancer cells or cancer DNA fragments. It is non-invasive, and it's real-time monitoring, and it's easy to repeat. It's like checking tumor fingerprints in the bloodstream. There are several benefits of liquid biopsy. It can provide early detection of cancer without seeing the symptoms of the patients. And while the patient is taking treatment, we can evaluate the response of the treatment. And if we face any resistance to the treatment, we can reach the resistance genes with this way. And liquid biopsy allows us to detect tumor progression earlier than the radiologic imaging. It can be repeated as often as a simple blood test, and it provides information about cancer heterogeneity. These innovations caused the rise of precision medicine, and the treatments of targeted therapies are mentioned as miracle pills. Over the years, the number of targeted treatments increased, especially after the 2000, many drugs were investigated. And the first FDA-approved drugs are trastuzumab and imatinib. You are seeing several different treatments, and the targets are used. For example, trastuzumab for HER2, imatinib is for BCR-ABL fusion, and sorafenib, palbociclib, many others. And these are some other treatments that we use in clinics, and we use the treatments in many hematological or solid tumors. I would like to mention two cancers, how it changed with the precision medicine. This is a lung adenocarcinoma. 20 years ago, we were treating the cancers nearly all the same, but now we are working NGS at the beginning of a metastatic lung cancer, and we may find many different targets, and then all different targets are treated differently. For example, If EGFR is positive, then these drugs are used. For example, osimertinib is very helpful for EGFR mutation. For ALK, lorlatinib improves survival a lot. And the other molecules, MET, HER2, Viorel, PIKs, 3CA, and many others, are specific targets that can be used specifically for different treatments. And the second example is breast cancer. With molecular profiling, breast cancer are divided mainly 5 different diseases. These are basal-like, Claudin low, HER2 enriched, or luminal A, luminal B. All are treated differently. For example, if basal-like or Claudin low breast cancers are treated by chemotherapy, the HER2 enriched treatments are treated mainly by target therapy, and luminal tumors are treated by hormonal therapy. And the prognosis of these patients are so different. For example, the survival of luminal A is higher than the others. So the innovations in precision medicine bring a new paradigm in oncology, tumor-agnostic therapies. This approach uses the same drug to treat cancers in different organs, as long as they share the same mutation. This improves survival and marks a milestone in precision medicine. There are many different approved target tumor-agnostic therapies. These are some examples. For example, if MSI high is found, then we treat the same way the cancer, even it may be an endometrial cancer or gynecological cancer. And the other FDA-approved biomarkers are Antrak, BRAF, RET, or HER2 biomarkers. And I would like to mention just about immunotherapy. Precision medicine does not only mean targeting genetic mutations, it also means targeting the interaction between cancer and the immune system. There are striking developments in cancer treatment with immunotherapies. One example is checkpoint inhibitors. There are PD-L1 or PD-1 inhibitors, CTLA-4 inhibitors. The immunotherapies use the patient's immune system as a therapeutic weapon, and it is guided by biomarkers as predictors. For some cancers, PD-L1 is a good predictor, like lung cancer, and for some cancers, microsatellite instability, MSI is a good predictor, and for some others, TMB biomarker works better. Maybe immunotherapy doesn't work for all cancers, but if it works, then the response is durable. There are control mechanisms used to prevent healthy cells from being recognized as foreign by the immune system. Cancer hides from the immune system by using these control mechanisms. We call it break. Checkpoint inhibitors remove these breaks and boost the immune system. Then the immune system can recognize the cancer cell and T cells attack and destroy the tumors. There are several different immunotherapy drugs. Besides checkpoint inhibitors, there are some cellular therapies. We use CAR T cells, TILs, tumor-infiltrating lymphocytes, cytokines, or tumor vaccines like T-Reg. And they are used in several different cancers successfully. I would like to mention about CAR T cells. It's a new model of cellular immunotherapy and very successful, especially in hematological malignancies. Excuse me. The CAR T cells are used successfully in AL, aggressive lymphomas, multiple myelomas, and they reduce the need for bone marrow transplantation. And they are not routinely used in solid tumors because there are some challenges for solid tumors. Patients' T-cell lymphocytes are genetically reprogrammed. A chimeric antigen receptor treated by tumor is added to T-cells, and then T-cells recognizes tumor antigens. The re-educated immune system destroys the tumors successfully. The impact of targeted therapies or immunotherapies are very huge. The patients who once had only months to live are now living years because of targeted therapies. These treatments don't just extend survival, but also improves the quality of life. This graph shows the survival gains by cancer type. The yellow bars shows the survival before targeted therapies or immunotherapies age, and the blue bars show after these treatments. And there are huge differences between survivals, especially in CML, HER2-positive breast cancer, and especially ALK-positive non-small cell lung cancer and melanomas. You see, after using the targeted therapies or immunotherapies, the survival differs so much. There are some challenges of precision medicine. Cancer biology is complex and constantly changing. When the drug is working in the beginning of the cancer, for a while it may not work because the cancer may change its targets. And genomic tests and targeted therapies are expensive, it's difficult to reach. And they are not equally available everywhere. These challenges are also opportunities for collaboration, working like the scientists like you. And these challenges may cause some obstacles for implementation in oncology clinics. The level of evidence for the genetic alteration is not definite yet for some alterations, and we don't know clinical benefits of some alterations, and we are not sure for the predictive value of biomarkers. They are still improving. When we make a NGS test, you may find several different genetic alterations, and most of them are not druggable, unfortunately. Even if they are druggable, they may not be available in the market. And the cost and reimbursement issues are still our concern. And there are also some concerns on inconsistent standardization and quality of testing. Now I would like to go future perspectives. There are some tools that will help us a lot for improving precision medicine. One is artificial intelligence. It helps us understand enormous amounts of genetic data. And nanotechnology, I'm sure it may soon deliver drugs directly to tumors with incredible precision. You are the master of these topics, and collaboration with you will improve our clinical implementation. The discipline of cell and genetic engineering will develop. Hopefully, access to these innovations will expand worldwide. And I would like to underline these sentences. A new era has begun in oncology. Cancer care is becoming more individualized. Precision medicine is transforming oncology from empirical to guided therapy. The therapy is targeting genetic alterations and boosting the immune system with proven clinical benefit will contribute a lot to cancer care. And cancer will be treated more based on driver mutations, not organ sites. These are my references. Thank you very much for your attention.
Thank you for amazing presentation. It's a very nice direction, and hopefully the cancer patients will have good treatment in the future, and it will prolong their life. If somebody has some questions. I would like to ask a practical question. It's in Andalou, how type of immune cell therapy are you using, and which type of cancer?
Immune cell, especially as I mentioned before, CAR-T cells and tumor-infiltrating lymphocytes are used. I'm involved in solid tumors, and they are not used so much. Just tumor-infiltrating lymphocytes is used in malignant melanoma, and the CAR-T cells are used especially in leukemia and lymphomas and myelomas.
Thank you very much. Some other questions? Thank you very much.
Thank you.
Now I'm honored to ask Vladimir to present new topological transition in superconductor nanoarchitecture from Leibniz University.
Thank you, Chair. Good morning, ladies and gentlemen. It's really a great honor and pleasure for me to be here on this occasion of the 7th conference in this role. And I'd like to start with expression of my gratitude to the organizers, Professor Shontia and academician Tiginyanu. Now we both are absent, but I ask you to transfer my thanks to them. I will speak about a special aspect of nanoarchitectures, which is related to new properties which emerge due to complex geometries of these structures. And these complex geometries generate non-trivial topologies. This topic is really very broad. And again, I ask you, Mr. Chairman, to stop me at a certain moment when you consider that the time is over. And then I will be happy to continue after the talk. It's really very broad. Okay. We are now celebrating an International Year of Quantum Science and Technology on the occasion of the first publications of the modern version of theory in quantum physics, and together with my congratulations to you all on this occasion, I like to recall that there are three main roads over this 100 years which produced and are producing now novel, fascinating physical properties. I will briefly recall them. First is reducing the dimensionality. You know, quasi-one-dimensional quantum wires, quasi-zero-dimensional quantum dots, quasi-one-dimensional quantum rings, and just this August, the third edition of my book, Physics of Quantum Rings, was published in Springer Nature. This shows one avenue, but now I have no time to speak about that. In the afternoon at 3 o'clock, I will be given another lecture on quantum rings in the Academy of Sciences of Moldova. It's open. I cordially invite you all to that other lecture. But the second direction is reducing of thickness of 2D structures down to single layer. It produced new properties like in graphene, graphene, transition metal, dichalcogenides, and a lot of other materials which certainly are being intensively investigated also for sensing and for applications, including in biomedicine. And the third direction, more recently, the way was to spread 1D and 2D manifolds into 3D. To be specific, imagine you have a film. Film is 2D. But if you roll it up, you will get something which is already in 3D, but it's not a three-dimensional object. And such objects are called nanoarchitectures. I will speak about this third direction today. Examples are here: nanohelices, nanocubes, and certainly the occurrence of this third avenue was possible due to the advancements of fabrication techniques. So, you will see what is it about, but here some overview of those methods which in fact allowed us to speak about such nice structures. classical ways of fabricating them are direct writing. It's indicated here. You have focused electron or ion beam in some chamber. These precursors, which under the influence of this beam undergo some chemical reactions and are sedimentated. But you can change the position of the focus, and if you rotate it, then you will get nanohelix, for example, like it is shown here. And another direction is self-rolling. It is so very clear and strategically important. A few words. Imagine you have two crystalline films in contact. They are of different materials and have different crystal lattice constants. And you have a third layer, which is white here. I hope you can see this white layer. And this white layer is sacrificial layer. Sacrificial. It is etched out from its position, and then two ultra-thin, sometimes it's even monomolecular, monoatomic layer, occur in situation when they can adjust their crystal lattices, but due to that a strain develops. This strain should be released, and under some conditions it is released by a deformation of rolling, as shown here. You see, in this way you can fabricate nanotube almost of any material. You know carbon nanotubes, but nanotubes can be produced with this method almost of all materials except brittle materials. There were attempts to make it of ceramics, of high TC superconductors. It does not go, it is not stable. But of other materials, metals, polymers, semiconductors, superconductors, they all produce a family of such nanomaterials. And especially for this conference, I like to dwell on two relatively new methods which are related to biology directly. It's the way how biology helps to have advancement in physics, just in this direction. DNA assembled superconducting architectures. By selecting DNA combinations, one can produce on demand some structures in space. Then these structures can be used as scaffolds for imposing there some metals like niobium, and in this case you will obtain a nanostructure based on that DNA, but the nanostructure itself is metallic or superconducting. It's at the early stage of its development, but it's remarkable. And these are some details. Unfortunately, I have no time. If you are interested, I will tell a sequence of stages which leave us with a nanoarchitecture. But even more recent and more surprising thing. This year, together with Professor Vasishta, we organized an international special session at the International Conference on Nano Science in Washington, D.C. And this is one of the talks which was presented there a couple of months ago. one can fabricate, whatever you like, nanostructures using bacteria. And it is shown here that niobium chloride salt in an anaerobic chamber with an actively growing methanogen syntropic culture. It's a kind of bacteria. These microbes are taken from soil. Here its names, you see here 4 names, I will not reproduce them, you can read them. Under oxidation of niobium chloride to niobium oxide, it's possible to get microbial fermentation of glucose, lipid and protein substrate molecules to produce different gases. And one of them is acetate, another is hydrogen, carbon dioxide. And all these gases and intermediate substances are consumed by this bacteria, which release methane. And this methane carburizes niobium oxide to niobium carbon. And niobium carbon is already a solid substance. And in this way, this produces flexoelectric systems which can be considered as nanogenerators. I especially dwelled on this specific topic in order to show you that the experimental advances are really remarkable in the field. But then, how to understand what we have and how to use that. I will dwell here on superconductors nanoarchitectures only because of restricted volume. And here are some examples like nanohelices, nanobridges, some whole nanowires. They are produced by different methods, but all of them are real. It's not a fantastic subject, it's a real subject. When we consider an open nano tube, is shown here, I would like to attract your attention to one very important consequence of this complex geometry. And I think this is the only slide where I left a few formulas, but there will be no more formulas. If we put such a superconductor tube into magnetic field here, then what happens? In a superconductor object which is in a magnetic field, there should be Meissner current, current which prevents magnetic field from penetrating the structure. But here we have an upper part of a tube, you see dark red, and a lower part is pink. We have already not one loop of circulating superconducting current, but 2 loops. And 2 loops is topologically absolutely different system from 1 loop. Already in this very simple case, we show that topology might be more complicated, and there will be other samples, but this is most important. And another story, that it's important which is the normal component of the magnetic field in order to develop superconducting properties. But normal component is easy to understand from this. Normal to the surface component is modulated on the diameter of this tube, simply sine function. And this modulation on the scale of 1 micron or still less what is achieved, it's remarkable. It's extremely difficult to make such a modulation in a planar structure at the scale of 1 micron. But here it is automatic, simply because we have rolled up this tube. And as a result, we have the following. Left part is a surface of a tube, and right part is a planar membrane. I think it will work. You can see. Yes. As distinct from a planar membrane, if vortices occur, superconducting vortices occur, they are in both parts of the tube, in upper part and in the lower part. But they move in opposite direction. And this is remarkable. In the same structure, there are already two motions of vortices. Why is it important? And I will go further. Here. There are two typical times. Time of flight and time between two subsequent nucleations of a vortex at the next. And these times, as shown in this panel, are strongly dependent on the radius. You see there are same curves for two times, but they are strongly dependent on the radius. And here in the inset, I think the key panel, take a square, some part of a planar niobium, and roll it up. It's the same material, it's the same object, but once it's planar, and another case, it's rolled up. And because of this transformation, I repeat, the same material and the same object, we change the physical characteristics, the typical magnetic field for studying vortex dynamics, by a factor of 3. When we get at this point, we understood that the majority of efforts in modern physics, both experimental and theoretical in what concerns nanostructures, are related to materials. We look for materials with non-trivial band structure. But we can have trivial materials. And by geometric transformation, we get non-trivial topological effects. And this is something to start with. But because of time, I will jump. When you increase current flowing in the tube from external source, when you enhance magnetic field, this same situation becomes more interesting. You get chains of vortices here, one chain, two chains, and at a certain moment even suppression of superconductivity. This blue part shows that there is almost no superconductivity. But when we looked into the phase portrait here, we observed that it's very interesting place on a line which is opposite to the car. The vortices and anti-vortices are spontaneously generated, and they move with very high speed. And in spite of the fact that a vortex which meets an anti-vortex from the next pair, they annihilate, but they move with such a high speed that the result, the induced voltage, shows a peak by a factor of 5. I like to transfer to you my admiration with this fact. This is one material, relatively simple, straightforward to produce. And you can increase its induced voltage by a factor of 5. All these effects are really very impressive. And there are different topological states. These, which I have just named, are called phase slips. There are also individual vortices, and we have combinations. I think colleagues, experimentalists here will notice it, that we can have in the same object both phase slips and vortices. This is something which is specific for this rolled-up structure. Because of possibility to have phase slip regime and vortices, we have two energy levels with hysteresis, so the voltage can be controlled by the transport current, but if you decrease current, it goes over another curve with hysteresis. And this is an experiment. People in Spain made this nanohelix by direct writing. Don't know whether it can be seen, but the diameter here, 277 nanometers. It's far below 1 micron. And the thickness of line is somewhere 30 nanometers. This is really a domain of nanostructures. Such a nanohelix revealed a few steps in the resistance as a function of current. It was measured, but the origin of these steps was absolutely enigmatic. We did not know what is it. And after some efforts, some calculations, we understood that the occurrence of this plateau is related to the development of phase slips, which I mentioned, in different half windings. So this helix is not uniform. And first, when you increase current, phase slips appear in two halves only. So this part is superconducting, but this is almost normal. Superconductivity is suppressed. So we have already some resistance. This is stage number at value 1. When we go to high values of current, 2 more half windings go into normal state. And finally, we finish our superconductor to normal transition, but it's not a monotonous. It's a line with steps. And secondly, these steps also give some idea of quantum nature of phenomena, because every vortex is a quantum of magnetic flux. But in this field of nanoarchitectures, I go further to absolutely new work with our colleagues at Max Planck Institute, who created a bridge. This bridge is shown here. So, and this line is 1 micron. Again, the sizes are in sub-micron regime. And there are 4 legs, and 2 legs are used for current, 2 legs are used for measuring induced voltage. But they are separated in space, and if due to the application of current, there occur vortices here in the red part. You see red part. The vortices move and under Magnus force to another part, and the measured values of the induced voltage, as distinct to previous case, they are not local. They are measured in different places. And this was confirmed by calculations which show that in these legs we have already a deformation of vortices, so in the sense they are relatives to Abrikosov vortices, but they are specific for such structures by all their properties, including their shape. So, if we add AC current, we can have a cornucopia of stages, including almost all superconducting here, almost all normal, but at a certain frequency we have a combination of both stages with very complicated Fourier spectrum. So we can influence the Fourier spectra of the response. We observed in calculations frequency locking at a certain interval of frequencies. I think this is the same effect which in normal body allows to support the frequency of heartbeats at a certain value. So if they are a little bit above or lower the system, body, due to frequency locking, returns the frequency of pulse to the normal state. This is certainly another scale. It's all in gigahertz, but the physics is essentially the same. And maybe approaching the end, I still like to tell about a couple of very interesting and recently considered phenomena. We calculated the spectra of the induced voltage in nanotubes, which occur due to the internal excitation. So we are interested what happens to vortices in this nanotube, for example. And it occurs that there are extremely rich spectra which are accessible in principle by method of spectroscopy. There are harmonics of main frequency expected, but there are also some harmonics which are not integer. And the occurrence of these non-integer harmonics, which are needed for electronics, you should have some frequencies, the more the better. They are due to such nice effects which probably you will be able to see. This is one chain of vortices, okay? But here somewhere, it's already two chains, bifurcation. Vortices move over two different paths. But not only bifurcation, we observed here the certain conditions in the magnetic field. I think here trifurcation, then it's quadrifurcation. I especially looked into the vocabulary, it exists in English, quadrifurcation. But upper have no special names, they are multifurcations. And they can be observed by spectroscopical regions. Then we can also include pinning sensors which can strongly influence dynamic of vortices and therefore the generated voltage or induced resistance. The shaping is also important. We should not make a full tube. We can have only a part, a segment of cylinder. It will also work because of the curvature. And here I start some fascinating discoveries concerning quantum rings, which I will be happy to tell to those of you who will attend my second lecture in the Academy of Sciences. At this point, I like to refer to a few perspectives, for example, for bolometry. Such helices which I mentioned, they have extremely high qualities as bolometers because noise is reduced. And here I list my collaborations, and among acknowledgements I like to mention European cooperation in science and technology, which joined together specialists from 30 European countries, and Republic of Moldova also a part of this. And our National Agency for Research and Development, which available this participation by the project Rebrain, for which I am grateful to this agency, and I am also grateful to you for your attention.
Thank you very much. Thank you for a wonderful presentation, and I ask people to go in the afternoon and to be present at the second part of presentation. As I understood, you'll be close to this direction. Thank you very much, and if somebody has some questions, please. No questions? Please, Professor.
From your experience, what would you suggest is the most promising material now which is not yet rolled up and you would like to roll up? So this is an experimentalist question.
I think in what concerns this superconducting real, the necessity is to enhance the working temperature. We started with an attempt just to take high TC ceramics, and due to their property that they are brittle, it was not possible. But there are other materials which have higher critical temperature than niobium or aluminum. But they are not brittle because they are crystalline, for example, or quasi-metallic. I can recall now magnesium diboride, for example. It has some higher critical temperature than traditional, conventional superconductors. But this technology of fabrication, though I represented it in one slide, is in fact based on many know-how. I tell you as experimentalists that if you do not take care of these conditions, the release of strain will be not enrolling. You will have some deformation like washboard. And in this case, all these nice features simply cannot be seen. So answer, possibly high TC, but not absolutely high, but higher than conventional, but which are not brittle. Then it will be more attractive for the application.
Okay, so we should continue discussion in the coffee break, but we can form tubes from brittle materials as well, as we have this other template method.
Wonderful, this will be great, but they should have the same sizes, say 50 nanometers or less thickness, and radius which is less than 1 micron. Then it will be okay. We also tried, by the way, to use the electron deposition methods in the Tiginyanu group to use them, but they were good for transport effects, but not for such superconducting applications. The work is going on.
Other questions? Thank you very much. Thank you for all audience being here at this session. And at 11:10 starts the poster session. Please be present there too. Now, coffee break. Thank you.
Yes.